Okay, let me go ahead and get started. Actually, let me get out of this. I'm going to give an introduction. All right. Okay, so I am so pleased that today we have Dr. Hongzhe Li speaking with us. For those of you who don't know Hongzhe, he's a professor of biostatistics, epidemiology, and informatics. He's vice chair of research integration. He's director of the Center of Statistics and Big Data, and former chair of the graduate program in biostats. He is also professor of statistics and data science at the Wharton School. He's been elected as a fellow to many places, including Institute of Mathematical Statistics, American Association for Advancement of Science, and American Statistical Association. His research focuses on developing statistical and computational methods for analysis of large-scale genetic, genomics, and metagenomics data and theory on high-dimensional statistics. So with that, I will let you take it from here, Hongzhe.
Great. Thank you very much, Sarah, for the introduction. It's really great to be here. So I figure what I'm going to do today is just to present some of the method that we have developed in the last couple of years in analyzing the microbiome shotgun metagenomic sequencing data. I also give you a couple examples just to tell you what those methods can tell us about the role of microbiome in disease, particularly in the inflammatory bowel disease. So as we know, in human gut, there are billions of microorganisms, and altogether they form the gut microbiome. And those gut microbiome plays a very important role in human health, including the protection against pathogens, immune system development, and particularly microbiome can produce a lot of really important small molecules, secondary metabolites, such as short-chain fatty acid, by fermentation of the dietary fiber. And in the last 20 years, there are a lot of papers talk about some type of association between microbiome and human diseases. Particularly at Penn, we spend a lot of time to look at the gut microbiome and inflammatory bowel disease. We also have some studies talk about look at the association between microbiome and the cardiovascular disease. So the tools, the really important tool for studying human microbiome is this shotgun metagenomic sequencing. So the idea is very simple. If you have a bio sample, for example, this could be the colon mucosa, or it could be the fecal human fecal sample, then you can just extract the DNA, then you do the whole shotgun metagenomic sequencing. For a given sample, usually we have a million, tens of millions, very short sequencing reads, and those reads can be aligned to some of the existing microbial genome database to give us some idea about the taxonomic profile of a given community or functional profile in terms of the genes that those microorganisms can provide. So the microbiome data analysis problem, it is a big data problem. As I mentioned, that usually for one given sample, we have tens of millions of sequencing reads. They're typically a couple hundred letters long. And those reads usually are sampled from a mix of hundreds of different bacterial species with a different but abundance in a given community. And in order to understand those sequencing reads, we need to look at the reference database. For example, in the NCBI database, there are over 300,000 bacterial genomes, and each genome in average is about 5 million letters long. So I'm going to give you 3 examples in my talk, just to talk about the role of statistics in analyzing those microbiome data. The first example is related to how to utilize the phylogenetic tree information when we look at the microbes. What you see here is a phylogenetic tree of the known bacterial species, and they are colored by the species that belong to different phyla. And when we have shotgun metagenomic data, the first things we need to decide is what particular gene we want to use to construct this phylogenetic tree. And in this paper, they use the 31 broadly conserved protein coding genes to build this sort of a consensus phylogenetic tree of the bacteria that we have their genome sequences. So after we have this tree, so when we build this tree, we don't really use the individual sample data. This is just a tree built based on the sequence of the bacterial species. But when we have individual sample data, there is now a very computationally efficient algorithm that can allow us to assign those reads, place those sequence reads, millions of reads to the branches of the tree. So this is just a cartoon example. The different color reads, different color line represent different sequence reads. And based on the makeup of the sequence reads, you can assign them to some position of the tree by calculating either the posterior probability of being a particular location of the tree or calculating some likelihood function. So now this can be done fairly efficiently. And suppose we have two microbial community, a red one, a green one, and after we place all the sequencing reads to the branches of the tree, we can just think about the data as a probability mass measured on the branches of the tree. And if you think this way, you can summarize the microbiome sequencing data as a structure like this. So you have some phylogenetic tree that is predefined. Then for a given sample, you can align the reads to different branches of the tree, and you can see some of the branches you have more reads than other branches. So you can think the data as a probability mass function measured on this phylogenetic tree.
So Hongzhe, can I just clarify? So the tree is you first determine the tree. Once that's determined, then you're saying, what are the communities that I have that I can place onto that tree? And the abundance is going to be reflected by the thickness width, of the line here.
Yes, the mass. Okay. How many reads, the percentage of the reads that can be assigned to different branches of the tree. Yeah. Okay. So that now is doable, computationally is doable for millions of reads. Okay. So that's the first step. Basically, after you obtain the data, you just summarize your data as a probability mass function on the tree. So the next question is, when you have two communities, often people want to define a distance, how close they are, how similar they are in terms of the microbial community, right? So the benefit
Dr. Tishkov, we're not hearing Dr. Lee.
I think his thing got frozen.
Yeah, I don't know if Sarah is also. Okay.
I'm sorry. This happened. I don't know what happened in my office. The internet sometimes get disconnected.
That's okay.
So
it was weird how it shut but it shut the whole Zoom down. I know that happened for everybody else, but like everything shut down and then it just like popped back up again.
Yeah, they said connecting. So I hope it's not going to happen too often. I'm sorry.
That's okay. It's not your fault. Well, that's all right. If we have to keep popping back on, we'll do it. No problem.
Yeah. So
I don't know if Dorothy, is it still being recorded or do we have to reset that? I don't know if she hears.
Yeah. I'll continue. So
yes,
we do need to record that, and I'm recording it. Okay.
Okay. Thank you.
Yeah, it's being recorded.
Yeah. So after you have the data aligned to the tree, the first things people often want to do is to measure the distance.
We don't see the slides.
Oh. Share. Yeah. So here's a slide. Okay. So one way to define the distance of the probability mass function of the tree is a very popular, very standard so-called Wasserstein distance. So I just tell you the idea of Wasserstein distance. The basic idea is you pick one location of the branches of the tree, and you look at the subtree below this particular location. Then you have for example, you have two microbial community, And their probability mass is denoted by probability P1 and P2. So the Wasserstein distance
I'm back.
basically it's a sum over all the branches of the tree. So this is summation over the edges of the tree, the length of the edge, and absolute value of the difference of the probability mass of the subtree under this branch. So it's not the difference of probability mass on each of the branches. It's a difference of the mass of the subtree below a certain branches. So that's basically is a Wasserstein distance. Okay, so you can see that biologically this is mean, I pick a Y, and anything below this Y maybe belong to a bacteria of certain at a certain taxonomic level. But you don't have to decide which level you want to look at the data. You look at all possible levels by various this branches Y. So that's so-called Wasserstein distance, theoretical distance. But once you have data, remember we don't really observe the true probability mass function. What we observe are the data measured from the sequencing data, and how to estimate this distance is actually not a trivial problem. So we published a paper in 2021, basically provide an close to unbiased estimate of the distance. I'm not going to talk about that, but that's the idea. So now after you have a distance, you have a way to compute the distance between two microbial communities. Then next question is you want to study association. For example, you want to ask whether the microbial association overall composition is related to some outcome, or whether some treatment can shift the microbial composition. And here we develop some kernel-based tools. Particularly, there's a software package we call the MIRACAT that can be used to test for overall association between microbiome and any of the possible outcome. And this method allows us to adjust for potential confounders. That's the benefit of doing this distance-based or kernel-based regression as compared to very popular Permanova type of test. So this is the test that you can use if the goal is to test the overall association between microbiome and some clinical phenotype, adjusting for possible covariate. But that's an overall test. But in practice, often you also want to ask, for example, when you compare the samples in the healthy individuals and samples from the individuals with inflammatory bowel disease, not only you want to know whether there's a difference in the composition of the bacteria, you also want to identify what are the difference right on this particular phylogenetic tree. So this is a so-called two-sample problem, and also you want to identify the main difference in the composition of the bacteria with respect to the structure of this particular tree. And this is back to the idea, so-called optimal flow idea I just mentioned, which is this delta E, remember, is a difference of the probability mass of the subtree under the edge E. And if this difference are the same between two groups, two populations, healthy and diseased populations, then there's no difference. Overall, there's no difference in the probability mass distribution. So essentially, we are testing whether this optimal flow defined relative to the structure of the tree is zero or not. And here we publish a method called active flow. Let me just tell you what is the idea of this test. So here is the data. So we look at this is a microbiome data from 18 healthy individual controls. This is actually mucosa, colon mucosal tissue microbiome, not fecal microbiome, and 14 individuals with active inflammatory bowel disease, active UC. And what you see here is a plot of the average difference of the probability mass of the subtree underneath each of the edge. So x-axis is the edge of the tree. The y-axis is the difference of the probability mass, average probability mass between the healthy individual and UC patients. And you can see the for many edges, the difference are very small. They're very, very small. They are close to zero, means there's no big difference. But in some part of the tree, you see a large difference in the probability mass between the healthy individual and the UC patients. So when you see something like this, you want to capture the difference. The most powerful or more powerful test statistics is by taking the maximum. Essentially, you take the maximum over all the edges, and you look at the difference of the probability mass of the subtree under that particular edge and normalize that by the variance. So this is like a typical two-sample test of the proportions. But the key is instead of you do the summation over all the edges, you take the maximum over all the edges. And this also allows us to identify the edges and the subtrees of this phylogenetic tree that give us the largest difference in terms of the probability mass between two groups, two populations. And this is more powerful than some of the global tests like PRIMANOVA or the MIRRORCAT test I mentioned under this sparse alternative. Sparse alternative basically said if you look at the tree, the probability mass over most of the subtrees are similar, but only in some of the subtree there's a big difference. Okay, so back to the back to the data example I mentioned. So here is a study we did at Penn. Again, this is the microbiome samples we look at are the colon biopsy samples, and we have 3 different groups. One is the healthy, group A is 18 healthy individuals. Group D are the UC patients, but they were not under treatment. And group C are the UC patients, but they are under active treatment for the inflammation. And here is based on their data, based on the data we observed in those samples, we can build this phylogenetic tree and we can align the reads to the tree. Okay. Now, if you do, go here. If you do, for example, the typical global test like a PRIMANOVA type of test to compare the overall microbiome composition difference, this is a p-value table. You actually do not see any difference among the groups A and B and C, B and C. But if you use our method called active flow, you have a very significant p-value when you compare the group A, which is the healthy individuals, and the group B are the UC patients. And it's a p-value is very significant. And why this is the case, this is exactly that the plot I showed you, you remember, for over many edges, if you look at the subtree below the edges, their probability mass are very similar. The difference are very close to zero. But when you do the summation, this PRIMANOVA test is a sum over all the difference. But that introduces a lot of noise. But if you take a max over the edges, you actually can magnify the difference, the signals. So that's basically the ideas. So this also points out to a very small part of the tree that include 31 OTUs, then belong to a family, a Ruminococcaceae family. The bacteria belong to this family. Then we identify there's a difference in the probability distribution in this particular subtree. And you can see the difference. See here, for example, in the healthy individuals, this is small samples at 18 people here. You can see their relative abundance is not zero. And in the UC patients, most of them, they don't carry this bacteria or with a very low abundance. But after you treat them with anti-inflammation drugs, you can see in this group, this bacteria abundance are increased. So this bacteria actually is quite interesting. What is really interesting about this bacteria is when you look at the bile acid produced by the liver. So the bile acid produced by liver, they are funneled into human colon. And at the colon, the bacteria like this Rumicococcaceae, they can turn those bile acid into the secondary bile acid. And only the secondary bile acid, they have this anti-inflammatory properties. So without this particular bacteria, this class of bacteria, this bile acid cannot be entered into their second phase of life. Therefore, because those UC patients, they lack this group of bacteria, so they don't have this anti-inflammatory properties. But after you treat them, some of the bacteria abundance increase. Therefore, you see this anti-inflammatory properties, and you see some a reduction of the inflammation. So this is I think it's a simple example to demonstrate sometimes when you use a better, more powerful statistical test, you make something more interesting than the tradition.
Can I ask you something?
Yeah.
So control is A, is that right? Unexposed is B, exposed is C. But you only saw a difference, statistical difference between A and B, not either with C. Is that correct?
Right. Because they're only, you see that C, C are the patients who are treated with the drugs, but it's still many of them, you don't see them carrying.
It's just because there's so few that.
Yeah.
Okay.
Yeah. And then this is different samples. Those are the cross-sectional 3 different samples. It's not the same group you treated and not treat them. Yeah.
Okay.
Yeah. So for a small sample size, I think this could be more powerful. So recently, we also extend this idea to more general continuous outcome. So not just a simple 2-group comparison. And we also have a way to adjust for confounders, potential confounding factors. So this is the first example. Basically, I present some of the tests to test the association between microbial community and some of the outcome groups. The first example. The second example I want to talk about is this shotgun metagenomic sequencing data actually also provide information on estimating the bacterial growth rate. Even we only have cross-sectional data, and it's possible to estimate bacterial growth rate. And just say if the bacteria community, a particular bacterial community, or a given bacteria are actually dividing, growing, usually they go through this exponential growth phase, where in the range about 90 minutes to 300 minutes phase, and you see the log of the number of the bacteria is linear. So they go through this exponential growth phase. So how are we going to estimate the growth rate based on shotgun metagenomic data? Remember, most of the bacterial genomes are those type of a circular genome. And in order for the bacteria to replicate, first they have to replicate their DNA. And the way that bacteria replicate DNA is by this bidirectional replication of the DNAs. So you have a particular location in their genomes, so called replication origin, and you have terminus region. So if this particular bacteria is actively dividing, then you tend to see more new DNAs in the region that is close to the replication origin than the regions than the terminus region, right? So based on this idea, in 2015, Koran et al., this Israel group, they published a paper in Science. Actually, they use this idea to estimate the bacterial growth rate. So the idea is simple. Is, for example, this is one microbial communities. You have a mixture of many bacteria, and some of them are actually dividing, and you have this shotgun metagenomic sequencing reads. Suppose you can align those reads back to the reference genome, their genome, bacterial genomes. Then for the bacteria that are actually dividing, you tend to see more reads coverage in this replication origin than the terminus region, right? Because you tend to see more DNAs in that particular region. But if the bacteria is not growing or is really not growing, then you expect to see sort of a uniform coverage of the sequencing reads over the genome. So they use this peak coverage over the trough coverage ratio as a way to quantify the bacteria growth rate, right? So, but this idea only works for the bacteria where we have a complete genome sequence, right? But nowadays, remember I mentioned that sometimes there are over 40 or 50% of the sequencing reads that cannot be aligned to the current known bacterial genome sequence. So now in this field, the people started to do the metagenome assembly. You can try to assemble the genome to construct something called metagenome assembled genome called MAGs. So MAGs basically represent potentially new genomes of a new bacteria. Okay. And now there's actually a really good algorithm available that allow you to perform genome assembly based on the shotgun metagenomic sequencing data. But the assembled genome, if you look at the assembled genome and the complete genome sequence, they are not the same. One thing that are different is, assembled genome is just a collection of contigs. We don't know the order of the contigs, right? Because there are gaps. So if you use the same idea, if you align the sequencing reads to those MAGs, to this recovered genome, assembled genomes, you cannot order those relative to the location of the genome because we don't know their location, right? So for example, if this is another data set, this is a real data set.
Wait, can we go back, Hongzhe?
Yeah.
Could there be a potential, I'm thinking about like our data set from Africa or people who are looking at ethnically diverse populations where there may be completely novel, there's going to be a lot of novel sequences. In fact, I think in our first estimate, I think like almost 80%, like we're not mapping or something. But then if that's the case, when you try to, you have to annotate it, map it back to some reference, right? Or, I mean, could there be a bias, like if there's more mutations in certain genes or regions, or I'm just wondering if there could be some kind of a bias that happens.
No, at that particular scale, like you mean the SNP, the SNP scale sequence difference, how they going to affect the assembly? I'm not sure. That's a good question for that particular sample. I remember when you do the contig construction, you have to allow for some of the mismatches. And after you have this beans assembled genome, you can try to see which you can try to annotate them, but most of the time you can only annotate them to the order of like a family order, sometimes completely unknown. Yeah. So back to the point I want to make is, so here is one of the MAG, one of this assembled genome. Okay. Then I have 6 samples. So on the top are the healthy individuals. On the bottom are the 3 samples from the IBD patients. And what you see here, the y-axis is a normalized log of the sequencing coverage, and each dot represents one of the contigs. So we normalize, we are taking into account the difference of the GC content and normalized by sequencing and everything after that normalization. And you plot the coverage over the contig label. And you don't see any trend, right? You don't see any like monotone type of coverage, right, in all those samples. But the key is this. Suppose you put this data into a matrix, and each row represents one of the sample, and each column represents one of the contigs. Then you do a PC, you do a PCA analysis. Then you order the contigs by the first PC. Then immediately you see there's a non-monotone coverage. And clearly you can see there's some area you have higher coverage than other area. And from this, for example, you fit a regression line, and you can estimate the peak coverage and trough coverage, and use that to quantify the bacteria growth rate. And that's the key idea of this method. We call it DAMIC. It's a dynamic estimator of microbial community that we published a couple of years ago. And Scott Daniel actually implemented this as a part of the standard metagenomic sequencing pipeline now. As I said, the goal is that the key step of our method is this simple PCA analysis to recover the order of the contigs along the bacterial genomes. And that works really well. And later on, Sarah, this is Ron master dissertation. He actually studied some of the statistical properties, try to understand why this simple PCA really works, and under what condition we can really recover the order of the context. And that itself can be useful when you try to can provide additional information for the assembled genomes, because you sort of order the context based on the bacteria growth rate. So that's the idea. So let me tell you what we can gain by studying the bacterial growth rate. So here is one of the studies that we did a couple of years ago. This is look at the pediatric Crohn's disease patients. So what we have is 90 children with active CD Crohn's disease from the CHOP. And so we got the baseline microbiome measurement. And those kids, they were treated by two different treatments. One is this enteral diet therapy. Another is anti-TNF therapy. Then we observed the inflammation of the disease as measured by this fecal cow protection, collect the stool sample at week 1, 4, and 8. And so we try, and also in addition, we also have 26 children, healthy individual, and the sample we have is a fecal sample. Okay, so we try to understand what does this treatment do to the microbial community of those kids. So at the end of the 8 weeks of the treatment in the anti-TNF group, there are about 50% of the children responded to the treatment in terms of the reduction of FCP. In the enteral therapy diet group, there are 12 kids, 32% of them has a reduction in inflammation. So the original paper we published with Jim Lewis and Gary Wu is basically saying that as after the treatment, the microbial community of those kids, they move towards healthy individual. They look more closer to the healthy children after treatment.
I have a question.
Yeah.
So actually, was it additive in any way? Like, or like if you did, so it looks like there was a reduction with just diet. There was a reduction with just anti-TNF. And if you,
but only 12%, 32%.
Oh. But I mean, I guess, was anybody given both? Like if
No.
you did both? Okay.
No. Also, Sarah, the good point I want to point out, this is not randomized. This is really based on the physician's decision.
Mm-hmm.
Yeah, it's not randomized study. So there might be some compounding factors, okay. But we just treat them as we separately look at each of the diet group, look at the kids' microbiome, before treatment, after treatment. And what we observed is before treatment, they are really different from the healthy children. And after treatment, they're getting closer. Just overall composition getting closer.
And do you ever consider genetic ancestry?
I really like to get those information. We don't have the genetic data. Yeah. Okay. So now we use our algorithm program academic, and we can estimate the growth rate for about 270 species. So some of them are known species. Some of them are the assembled bacteria. And among the assembled bacteria, we found 7 of them. They have a very different growth rate between the healthy samples and the kids with Crohn's disease. And here I present those 4 different assembled genomes. And this gives you some idea about completeness of the genome assemblies. Contamination means context of one species may be assembled into context of other species. And for 6 of the 7 MACs, we see a higher replication rate, growth rate in the healthy individuals than the Crohn's disease patients. And those MACs you see here, most of them we don't know. This seems like they are the new species because based on the marker linkage, we can only assign them to, for example, phylum level or family order level. That's the best we can do based on the assembled genome. So they may represent new bacterial species. And you can see their estimated growth rate between the healthy individual and the Crohn's disease patient. They are very, very different. But what is interesting is if you look at the kids after treatment, the same group of the kids after treatment, you can see for the same bacteria, and their growth rate started to go up. So this is one is a baseline, 1 week, 4 week, 8 weeks, and their growth rate getting close to the healthy individuals for the first 2 bacteria. For the bacteria that with a higher growth rate in the patients, their growth rate started to go down. So we hypothesis that why there's a change of the bacteria abundance after treatment, maybe is due to different rate of growth of the bacteria, or maybe the treatment somehow lead to increase the growth rate for some of the bacteria. We don't know what's caused with, but this feature just give us another way to look at the microbial community, not just simply look at relative abundance of the bacteria. So this, yeah.
Getting that here, if any of these participants are also getting antimicrobial, because that's part of the standard management of patients with Crohn's disease, right? If they were getting antimicrobial, which may you keep mentioning about confounder, if that might have been a potential confounder, major one.
For this study, no. For this study, not, at least not simultaneously. So either either they have this enteral treatment, or they anti-TNF. Yeah. Confounding factors are really important to adjust. I agree with you. Yeah. Okay. So this is basically our algorithm dynamic, and as I mentioned, the key is to use a PCA to figure out the order of the contigs based on the uneven coverage of the reads of the sequencing reads, and that only works for the bacteria, for the community with bacteria that are actively divided, right? If the coverage is uniform over all the samples, then there's no way you can order them, right? In that case, you have like a flat line, then you cannot estimate the peak against the trough, right? So only for the community where in some of the samples that this particular bacteria are actively divided, then you can try to figure out what is the growth rate. Okay, so the last example is this idea of BGC. So I think the microbiome research is really moving from only look at the relative abundance of bacteria, composition of community, gene abundance, to function understanding of what do those microorganisms do to affect human health. So one way the bacteria can play important biological role is by generating those important bioactive secondary metabolites. And this includes many of the known antibiotics, anti-cancer reagents. And those secondary metabolites, they are coded by genes that cluster together in the genetic package. And this together is called the biosynthetic gene cluster. And here are the example of 4 of those BGCs, and they are derived from different bacterial species. So those are different bacterial species, and they code the different biosynthetic product of different types. And you can see those BGCs are different in terms of length, in terms of genes involved. However, you do see they share some common genes with some common functions. For example, they all carry genes that transport genes. They all carry genes of dehydratase genes. So basic idea is there are some similarity in terms of the the makeup of those gene clusters. So now the goal, so this is initial paper was published in Cells, and in the Cell paper, they propose to identify additional BGCs by using this two-state hidden Markov model. And this is publication from Mike Fishbach group at Stanford. So what do we have? Now there is a database called the MICROBIOME database where you can find the currently known BGCs. There are about over 2,000 known BGCs. Known means they are validated BGCs. And you go to this database, you see the structure of the BGCs, what are the genes involved, which bacteria carry this BGC, what is a biosynthetic class, and the compound they can produce. So there is this database available. So our goal is to do this. So now we have about 2,000 BGCs in this MICROBIOME database. And we have 6,000 bacteria species where we have their complete genome sequence. So our goal is, based on the known BGC, can we identify all the BGCs in the genomes of this 6,000 bacterial genomes and identify them and categorize them into broad BGC classes. And this is what we did. Basic idea is this. So you have known BGCs, you convert their DNA sequence into protein family domains. And you build, so you have the known BGCs, then you have to create some control non-BGCs by converting the reference genome sequence into a sequence of protein family domains. And you just basically create this pseudo non-BGCs by randomly pick some of the protein family domains. So that's how we build the training data set. So then what we did is this idea of, so we want to identify additional BGCs. So the key idea of this deep learning algorithm is we can utilize the name of the protein family domains. We can utilize the protein family clan information. So clan information basically tell us some of the protein family domain, they are similar in function, in structure, in the underlying hidden Markov model. And also each of the protein family domain, we also have a description of this protein family domain. And you think about if you have data like this, it's more like a text data. It's not a numerical data. It's very difficult to use the existing method, machine learning method, to make a prediction. So in this setting, I think a deep learning works pretty well. So basic idea is you can embed the name information, the similarity information, and function description information into a numerical vector for each of the PFAM, for each of the protein family domain. Then you can run this deep. I'll skip the details, but the rationale is this: is even the biosynthetic pathways for unknown compound family, unknown BGCs that are very different from the known secondary metabolites, from the known, but the secondary metabolites, they use the same broad enzyme family for their key reactions, right? So use this assumption. You can, so this is just some summary of the known BGCs, just to give you some ideas in terms of how many protein family domain they include. So most of them are pretty short, like 4 or 5 protein family domains. But some of the BGCs, they include hundreds of those PFAMs. And this is the idea of the clan information. Basically tell us some of the BGCs, some of the protein family domains, they are similar, and we can use that information in the learning algorithm. So anyway, then we build this deep learning algorithm, and I'll just tell you some of the results because time is up. So this deep learning algorithm, this embedded idea is, I think, is really powerful. What you see here is a Disney plot of each of the known BGC that belong to this 6 different classes, RAPP, polypeptide, saccharide, saccharide NRP, different broad biosynthetic classes. And this is what we, this is for the training data set. You can see this machine learning, this deep learning algorithm can indeed learn the difference among these 60 different broad classes of the BTCs. Right? Except for the yellow group belong to the other class. So other class means, they may have multiple, they may belong to multiple function group. But for the other, you can separate them just based on their sequence structure. So, and similarly, you can separate the BTC, non-BTC pretty well. This is for the testing data set. And we can evaluate this by leave out some of the genomes with a known BTC, and we can create this pseudo genomes. And just to give you a little summary of what we can achieve in terms of the prediction. So we can predict all those BTCs and calculate the sensitivity, specificity of the predicted BTC and the known BTC. And this is F1 score statistics. And what we compare is our method. And there was prior to what we did, there's another paper published in the nucleic acid research. They have their algorithm with the first column. They also do some deep learning algorithm for the same problem. But for their algorithm, they only use the protein family name information. They didn't use description information. They didn't use similarity of some of the protein family domains. So you can see a fairly substantial improvement in prediction of the BTC using our new deep learning algorithm. And so, oops, keep doing this. Sorry. So at the end, we apply our algorithm and make a prediction, make a lot of predictions. So this is our algorithm. And this is another evaluation. So basically, remember, we build our model based on the known BTC in the MIBIG database version 1.4. And the new version 1.5, people deposit another 160 known BTCs. And we use our algorithm to see whether we can predict those known BTCs. And here is, again, the result in terms of F1 score. So F1 score is a score that summarizes both the sensitivity and the specificity of the prediction. You can see just for BTC, non-BTC prediction, the algorithm actually can achieve fairly high F1 score. And here, each row represents, so how to evaluate the BTC we predicted and the known BTCs? Because BTC, right, is a cluster of genes. So what we did is to look at the predicted BTC and known BTC and see how much they overlap. They overlap by 20%, 40%, or 100%. Even the 100% means the BTC we predicted is exactly the same as the known BTC. We can achieve an F1 score of 85, 88%. However, the BTC class is a little difficult. So the F1 score can be 70 to 55%. And the reason is the sample size is a little small. So here's just give you some idea about in our training data set, for some of the BTC classes, the number of samples, the number of BTC that belong to a particular BTC class is small. So it's hard to make a prediction. There is one class, which is this IRPP class. Although the training sample, the number of BTCs of this particular class is small, but we actually make a lot of predictions. And the reason is, if you look at the BTCs of this particular class, they are very similar. They are very, very similar. Therefore, it's very easy to identify those guys. So our deep learning algorithm actually identify quite few BTCs that belong to this particular class. And just to give you some ideas, so eventually we cross about 6,000 bacterial genomes. We predict about 170,000 BTCs. It seems like a big numbers, but just look at like the coverage of the genome, those BGC, roughly they cover about 10 to, most of them cover about 10 or 20% of the genome. So it's not like we predict everywhere of the genome they carry this BGC. And in terms of the number of genes involved, is also a small number of genes involved in those BGCs. But now we're at this stage. It's really hard. If you ask me how sure those predictive BGCs are, is very difficult to answer that question. So at currently, we build a database. Basically, we have some idea of bacteria of a particular phylum, how likely they can produce certain class of the BGCs. So this might be useful when we try to quantify, for example, the biosynthetic function of a given microbial community. So that's the goal of next step of our research, is really try to give me the structural metagenomic sequencing data. I really want to get some idea about what are the potential biosynthetic product in the broad class sense that this community can produce, and look at the difference. So anyway, so this is the third example. Basically, we have now this deep learning algorithm that can be used to predict the BGCs in the bacterial genome, and compared to the other method, our method can also predict a broad BGC class. And we are building this database. Hopefully this can be useful. So I think my time is up. I just want to conclude my talk. Basically, what I presented today is just 3 different problems that we try to answer with a learning method or statistical method in microbiome data analysis. So currently, my main collaborators, they are really interested in studying nutrition, precision nutrition, the microbiome, and how they are related to host immunology and the health. So we're still working on some of the problems in this area. And finally, I just want to thank my postdoc and students who are working with me on those problems, and my long-term Penn collaborators, Gary Wu, Rick Butzman, Jim Lewis, Kyle Bittingers, and Tony Kai from the stat department. We work together on some of the more theoretical problems of the method. So that's all. Thank you so much for
Great.
listening.
Thank you. I know everybody's clapping. I can tell you, I personally learned a lot from that. Even if I didn't understand all the math, it was super helpful and informative for some of the things that we want to do. Does anybody have any particular questions? And I'm thinking, I don't know, Alex, do you want to comment at all about some of the things we're thinking of doing, and if these might be appropriate methods?
Sure. So the thing that most struck me was the first method that you described, which was ACT flow as kind of a replacement for Permanova.
Yeah.
I think one of the most important things that we're trying to figure out is just how these different metadata categories of individuals, whether they be ethnic groups or geographic-based groups, or just groups where maybe one group has high Prevotella and one group has low Prevotella. What else is kind of different between them? I was thinking of doing Permanova, but I think now we're going to
I mean, if the difference is only on some small part of the tree, this ACT flow can be more powerful, right? Because the other part, the Permanova essentially is that flow, you take, instead of taking max difference, you sum them all together. And that actually can introduce a lot of noise to the final statistics. That's why it's not as powerful. If the difference is so big, it's everywhere on the tree, but usually we don't observe that. So that's exactly the motivation of this this max type of statistics can be powerful under this sparse alternative. So,
Yeah,
so Sarah,
because
the data you have is a 16S or shotgun data?
so we have 16S from, I don't know, something like how many, Alex, 700 or more? We have like 600 or so in Cameroon, and then 100 and something from Tanzania and Botswana. Is
That
that
sounds
right?
right.
And then the metagenomic currently is how much?
About 180 individuals just from Cameroon.
And we're about to greatly expand that, I think, to over 400, if I recall, something like that.
Yeah,
Oh.
we were looking at something like 400 Cameroonians and maybe another 60 from Tanzania and Botswana, just
Right.
for comparison of subsistence strategy.
And then the other thing, Hongzhe, that we want to do actually is to integrate with the lipidomics data set that you've been working on with us. And then we also have the untargeted metabolomics data set from Chuck Barrant, and we want to try to integrate with that as well. So we would love to follow up with you on this.
Sarah, now we have quite a few studies that we collect both the microbiome and metabolomic data, both from the fecal sample and from the blood. But we don't have the data of your scale because usually we have small sample size in patient study. And you have genetic data.
I mean, that's we have
the Andy Clark, they look at the genetic of microbiome. I don't know. Have you actually tried to look at that in your?
Do you mean trying to infer, like they inferred the human genome sequence from the gut?
No.
Is that what you mean? Or
they study the effect of host human genetic variation on microbiome.
Right. That is our next goal. That's next on the list. We actually have a whole genome sequence data for most of these people. And that's exactly what we want to do and try to integrate it across multiple levels. Because one of the things that has been frustrating, and we would love to talk to you more about, is that we see so many unknowns, like so many in the African data set compared to what is observed in a typical US, European ancestry data set. And the other thing is that I've always been surprised that these groups that have dramatically different diets and dramatically different environments, we're not showing more markedly different, more differences in the bacteria. It was like, relative amount of this common bacteria, relative amount of this other common bacteria. And I know there has to be ways to dig in deeper.
Yeah, I mean, that's extremely rich data set to look at. I mean, even you just look at your data with some of the public available Caucasian gut micro. Yours is also fecal microbiome, right? Mm-hmm. Yeah, that itself, I think, is you can see a huge difference and try to figure out, what are the key unique bacteria to the African population.
Exactly. Well, I tell you what, we will, Alex and I will definitely be following up with you.
Yeah, I'll be really, and I'm really interested in.
Good. I'm glad to hear that.
Some of the data.
And that was super informative. It's after 4 o'clock now, so I guess we can go ahead and stop there. But certainly, we'll be following up with you. And thank you so much for that super informative talk.
Thank you. Thank you very much.
All right. Take care, everybody. Bye-bye.
Bye.